So I would like to welcome one of our esteemed board members who is going to do our formal opening for this evening. And could I please ask James Gray to come forward and do a formal opening for us. Thank you, James.
E ngā manuhiri, tēnā koutou. Nau mai haere mai ki AMRF Lecture Series 2. Whakatau mai ki runga i a mātou. He mihi tēnei ki ngā kai tautoko o tēnei kaupapa ko AUT University, ko Auckland University, me JMI Wealth, kia ora koutou katoa. Ngā mihi nui ki ngā kai kōrero o te pō, Dr. Rosemary Frey, Associate Professor Gary Cheong, me Professor John Windsor, nau mai. Ka huri atu te mihi ki a AMRF. Ngā kai whakahaere tēnei hui mō te rātou manaaki me te tautoko. Kei te mihi tuatahi te mātou nui o te rangi nāna ngei mea kātua. Tēnei te mihi ki a koutou. Ka nui te koa me te hari ki tūtaki i a koutou i te kaupapa. Nō reira, ngā mihi nui ki a koutou, tēnā koutou, tēnā koutou, tēnā koutou katoa. For those who don't know me, yes, I'm a trustee on the Auckland Medical Research Foundation. My name is James Gray, and on behalf of AMRF staff and the board, I'd like to welcome you all here this evening for our second lecture of the series. I'd also like to thank some of our sponsors, AUT, Auckland University and JMI Wealth. Small caveat, I also work for JMI Wealth. But thank you for their support. We'd also like to welcome our speakers tonight, Dr. Rosemary Frey, Associate Professor Gary Cheong and Professor John Windsor. Welcome to our lecture tonight. Also like to thank AMRF, our staff, for putting this together tonight. Looking forward to hearing hearing the speakers tonight, and as you are all here in person and online as well. So yeah, I'd just like to welcome everyone here tonight, and I hope you enjoy this evening. Thank you.
Well, thank you, James, for such a wonderful wonderful welcome and introduction. And I'd also like to warmly welcome you all here tonight. Thank you for coming out on a chilly night, braving the traffic. At least it's a lovely, fine night, so I'm glad you could all find your way here. So, as I introduced myself before, Sue Brewster, Executive Director of Auckland Medical Research Foundation, and this is the second in our 70th anniversary celebration speaker series. So, and we're also here tonight celebrating the awarding over $100 million of medical research funding. So there's a lot to celebrate. Like James, I would like to thank our 70th anniversary partners, who are JMI Wealth, represented by James Gray here tonight. I'd also like to thank the Auckland University of Technology, represented here by Brett Cohen, and also the University of Auckland, and we have Mark here tonight representing as well in place of Frank Bloomfield. So thank you to your partners. You've made a lot possible to happen this year, and you are the ones also for AUT and for UOA doing the research, so that's fantastic. And obviously JMI Wealth are helping us bring in the dollars. So obviously with both topics, being palliative care and assisted dying, there can be some sensitive material in these presentations, so I just wanted to make you all aware of that, and hopefully you all came expecting these topics to be covered.
Kia ora koutou anō. Welcome back after a brief stretching break. I am not Professor John Windsor. I am Tim Angeli Gordon, but it is my distinct honor tonight to get to introduce Professor Windsor. He didn't know I was introducing him, and he asked me to be kind He also asked me if I had his bio, which, full disclosure, I have made up this bio. So, but one of the things I love about these sort of nights is getting to meet the people as well and getting a bit of an insight into who they are, not only their research, but also who they are. And I think we had a wonderful window into those first two speakers. So I will try and also give us a bit of a window to Professor John Windsor. One of the things as academics, we hear people's degrees or titles, and we joke that those are letters after your name. So as you get a degree, you typically acknowledge that by putting the letters after someone's name, and it tells qualifications of people. I realized there were too many to read, but I also thought to myself, I said, I wonder if there's more letters after Professor Windsor's name than are in the alphabet. So are there more than 26? He asked me to be kind, so I won't belabor the point, but there are 34, and those are just the ones I've found, so there's probably a lot more than that. And I don't say that to put him on a pedestal necessarily, although he deserves it, but because that tells me a few things about who he is. One, he loves learning. Two, he's always striving for excellence. And three, he's widely respected by his peers. And I think all of those are really admirable traits. So most of his peers and others would describe him as an academic surgeon. So that captures a couple of different facets. One is a surgeon, and he's a phenomenal surgeon. And the best way I can capture that is that in talking to his surgical colleagues, many of them have said that if they ever need to have surgery, they want Professor Windsor holding the knife. And I think that is the best compliment that you can have. Now, academically, he is also a prolific researcher. He's published well over 400 papers in some of the very, very top journals in the world. They've attracted over 30,000 citations. So a citation is when somebody else publishes a paper and refers to his work. Over 30,000 of those. But imagine, if you will, for a second, the number of people that have had conversations in the laboratory or around the clinical ward or around the surgical theater that have referred to Professor Windsor's work in their clinical practice or in their clinical care. I would dare say it's many, many higher than that 30,000, which is already a phenomenal metric. As if that's not enough, being both a surgeon and an academic, he's also a great innovator and an entrepreneur, which I think he'll touch on tonight. But you can look up all of that information on Google, which is part of how I created my own little bio for him here. But what you can't look up is, I think, what to me sets him apart, and that is his mentorship. He's a phenomenal mentor, and there's literally an entire generation of surgeons internationally who have been trained by Professor Windsor, as well as an entire generation of academic researchers. And I'm happy to put my hand up as one of those people who has benefited from his training as a researcher. We haven't spent a ton of time together, but in the time we have spent, he's clearly one of the most influential mentors. And the best way I can describe that and who he is as a person is every time we meet, I realized, when coming up with this little bio, that he asks me three things. He asks how I'm doing, he asks how my fauna is doing, and he asks how he can help me. And I think if we just think about that last one, every time we catch up, he asks how he can help me. That really, I think, sets him apart. It's something that we need a lot more of in academia, I will say, and something that is the highest compliment that I can give to who he is as a person. So I'd like now to welcome Professor Windsor to the stage.
I don't know what to say after that. Thank you, Tim. Can you hear me all right? I'm going to wander a little bit as well. Really kind words. Thank you, Tim. It's easy to mentor somebody like you. I want to spend a few minutes with some introductory comments, and then I want to go through 3 clinical observations, a new paradigm, 5 laboratory experiments quickly, not too scientifically, I hope, and then 2 translational treatment strategies which we're working on that comes out of this new paradigm. You'll have seen that talking about a forgotten highway and organ failure. I hope it will become apparent what the highway is. Should be obvious. Firstly, I want to thank Sue and her whole team, the trustees, those who review grant applications for their support. Two recent projects which have been supported by AMRF, which I'm perfectly involved in. I supervise Sang Ho at the top there, part of Kaitia Health, where they're doing intracranial monitoring of pressure for hydrocephalus, going really well. And then guess who? Tim, doing some great work on endoscopic mapping of gastric arrhythmias or gastric electrophysiology, and also looking at ways in which we might treat abnormalities of rhythm within the stomach. Exciting work. Now, my favorite topic, the pancreas. Pancreas often forgotten. In fact, early anatomists said this is just a fat pad upon which the stomach sits. Actually, it's a highly complex organ, and most surgeons don't want to touch it because it bites. There's two organs in one. It produces enzymes for digestion. It produces hormones for metabolic control. We don't really understand why they're co-located. That's a whole area to discuss at some other time. The anatomy is fascinating and challenging, and I chose the pancreas because it is surgically a very challenging organ. It's not in the abdomen, it's behind the abdomen. And it lies, as I like to say languidly, over the spinal cord, over the aorta, over major vessels. And so the dissection of that gland is complex and risky. Three common diseases, all of which have fascinated me. Acute pancreatitis is one of the most common gastrointestinal admissions. And we're going to focus on that tonight. Chronic pancreatitis, not the same disease, incredibly painful disease. People get depressed with it, and pain is really difficult to control. And then pancreatic cancer. It's going to be the second or third most common cancer death as we go forward. We are making some progress, but most of my pancreatic cancer surgery was in the end palliative. And I appreciated some of the comments tonight very much. So, the cause of death over centuries has changed. When you look at the list on the left, common diseases, in the old days, death used to occur as a direct result of the primary disease. We're not much better at caring for patients now, so death is most often actually not from the primary disease at all, but from what we might call secondary disease or organ failure, which is the topic I want to talk about tonight. And really, organ failure has been called the plague of modern medicine. The better we get at treating the primary disease, patients ultimately sometimes die, and this is what they die of, organ failure. And so here's a picture from the intensive care unit or Department of Critical Care Medicine in Auckland. You've got a patient who's anonymous on that. You can see a lot of things happening around that bedside. This is what it looks like when you're in organ failure. You can see a dialysis machine, kidney failure. You can see a ventilator, respiratory failure. You can see infusion pumps here for inotropes and other things like that, cardiovascular failure. You can see blood transfusion happening, intensive monitoring. This is what it looks like in intensive care. And when a patient gets into this state, there's a 20, 30, even 40% chance of dying still. Why is that? Well, we're very, very good at organ support while the disease takes its course. But people ultimately die of organ failure because we actually don't have a treatment for the organ failure. We support the physiology, but we don't have a treatment which addresses what drives organ failure. So, apologies for those who might be a little bit squeamish. Do look away if you need to. This is a rare picture of a fresh pancreas from a patient who died who also had acute pancreatitis, and I use it a lot in my teaching. Sorry, beg your pardon. So local problems in the pancreas can, in simple terms, be described as there's an inflammatory process going on, swelling and redness and that. But there's also a process called necrosis, where part of the gland actually dies. A failure of microcirculation, the premature activation of the pancreatic enzymes, which should be digesting the food in the duodenum and small bowel, gets activated in the pancreas, and the enzymes turn against itself and start to digest the gland itself. A really fascinating process. But there's not just local problems with acute pancreatitis, we also have organ failure. And so these are the three organ systems which fail most often, and I've mentioned them already: lungs, heart, kidney. And when you look at the pattern of organ failure in patients with acute pancreatitis and the pattern of mortality, it's been a bimodal distribution. There's early deaths, which we talk about as cytokine storm. All the inflammatory cytokines, the cascades just go out of control, and this seems to drive that early organ failure. It's not the only explanation, but that's what people say. And then we have a later organ failure that might come on after 3, 4, 5 weeks in a patient with severe acute pancreatitis. That's due to infection of the dead tissue within the pancreas. So infected pancreatic necrosis leading to organ failure. We are now much better at the treatment of infected pancreatic necrosis, and that second peak is pretty much flattened. So we're really dealing with early severe pancreatitis and organ failure in the first 1 or 2 weeks as the main problem. So the risk of organ failure and death in patients with severe acute pancreatitis is similar to what you would see with an acute coronary syndrome or a stroke. It's that serious. And when you come in the door with those diseases, there's immediate things that happen. You know, it's an emergency. Whereas if you come in with pancreatitis, we don't have anything to do like you would with a heart attack or a stroke. Okay, so in my clinical practice over 35 or 40 years with pancreatitis, there's 3 pretty barn door obvious observations which have struck me. They're not original to me. In fact, everybody could observe them if they looked. The first is that there is a stereotyped response to acute and critical illness. Now, irrespective of whether it started with meningitis or pneumonia or multi-trauma or severe hemorrhage, pancreatitis, what happens to the patient and what happens at, when you assess them at the bedside, it actually looks similar. You wouldn't know what the underlying trigger was or disease was in many, many cases. And you go from a process, as you see here, of systemic inflammatory response through to multiple organ dysfunction syndrome, and then to multi-organ failure and death. That sequence happens irrespective of the underlying disease. That's an important observation. And why is it important? Because it suggests if there's a common response, there must be a common mechanism. Secondly, and you've probably seen this in your visits to an intensive care unit, it seems that patients with acute and critical illness swell up. In fact, not in the earshot of patients, one hears the description, the Michelin Man, if you know the advertisement in the States. I mean, these patients can become incredibly edematous, and you can see that in various parts of the body and through a number of different processes, which we don't need to go into. And there are consequences of edema, and we now talk about the fluid accumulation syndrome affecting the organ function in all the systems. Gastrointestinal down the bottom there, but all the systems can be impacted by this edema. When I grew up in surgery, I was told by Professor David Carter in Edinburgh, when you're giving fluid therapy in acute pancreatitis, you just keep giving as much as you can until you start hearing creps in the lungs, which means, some respiratory failure. Just load them up. They need fluid. We now know that that kills patients, and there's an optimal amount of fluid that we should give. But our patients were often quite edematous, and I'm sure that contributed to poor outcomes. The third clinical observation one might see in radiology, where a patient with acute or critical illness, acute pancreatitis, may get a distended abdomen. They may not be able to pass wind. They might have high volume of aspirates or vomiting because the bowel's just not working. And that's what we call ileus, which you see here. If you do a CT scan, you might see gas in the wall. That's gas in the wall of the bowel, which you can see histologically here as little bubbles. That can resolve, but it's indication of ischemia of the lining of the bowel, if not ischemia of the whole thickness of the bowel, which you see here. If we open up a patient with severe ischemia of the bowel, it looks like that. You flick the bowel in theatre, it doesn't respond, it doesn't contract. It's distended, a little bit smelly, and you might in fact encounter perforation of the bowel. Quite a severe problem, non-occlusive mesenteric ischemia. So we got pretty interested in the gut. And I grew up in India in the Himalayas as a child and was very aware of the caste system, and in my own mind, I've always thought about the gut being considered a second-class citizen, and in one sense it is, because there are vital organs. The gut is not one of them. You know, for immediately required for life, you need heart and the lungs for sure, kidneys, probably not the brain. In fact, you can live without brain function. Not very high quality of life. But what happens in acute and critical illness, especially when blood is required to vital organs, where does the blood come from? Where does that extra supply come from? It comes by vasoconstricting, contracting the vessels supplying the bowel. What does that do? Well, it causes ischemia of the bowel, but we're prepared in our body, our physiology, to sacrifice our gut for the sake of the function and perfusion of vital organs. So I mentioned non-occlusive mesenteric ischemia. Infarction means death, and we can have different diseases causing a bit of redness here. Ultimately, that duskiness and death. You can lose the whole bowel, full thickness, infarction, perforation, peritonitis, multi-organ failure. And so this process can be subclinical. That is, all that you see is a bit of distension, maybe a little bit of tenderness, and people don't seem to regard it as a serious problem. But there are definitely consequences of even mucosal ischemia of the bowel, and we'll come to that. I love this study. We did it in the mid-1990s with Professor Bonham's son, Martin, a Yugoslav who came to us as a refugee and Fikri from the Middle East, who gave 5 years of his life to our lab without asking for any money. And we did a number of, I think, important early studies. One of them was this. This is a catheter that's placed in the stomach, but it's got a balloon. Can you see the balloon with fluid in it, just saline in it? It's semi-permeable to CO2. So you can sample that saline and get a CO2 carbon dioxide measurement from that, and that gut is producing CO2. If you take a blood gas and take a bicarbonate from the blood gas, you can calculate from these two sources a pH. And we call it PHI, or intramucosal pH. There's some assumptions in there, but it will actually tell you how ischemic the gut lining is. This is the first study that showed that if you've got a PHI less than, sorry, not good at that. If you have a PHI of less than 7.25, you've got a high probability of death. So linking gut ischemia with death in pancreatitis was the first time that was done. The other thing that I think that's important to note, in our zealous attempts to treat organ failure and maintain organ function at all costs, we actually can further damage the gut. Even common things that we do, opiate analgesia, inotropes, fluids, antibiotics, enteral feeding, all those things can contribute to gut ischemia, gut injury. And you'll see why I think this is important. So, just to summarize, organ failure can cause gut injury, and gut injury, as I'll be showing you soon, can contribute to organ failure. That's a vicious cycle. And so gut injury and organ failure and gut injury. So other people obviously have been interested in the role of the gut, and various concepts or models have been developed. One of which is the gut motor concept, and many people have heard the word bacterial translocation. This means that if the gut wall is damaged, bacteria, which is in the, we have lots of bacteria in our gut, they can get out, they leak out through this damaged gut, they get into the blood supply and make people sick. The trouble is, people who've been measuring bacteria in the blood coming from the gut have not been able to prove this theory, at least in the context of multi-trauma. So there's some questions about that. Then there's the gut starter concept, which is that the gut's compromised by a lack of blood supply. How does the body respond? It sends in the troops, white cells, neutrophils, lymphocytes, monocytes, macrophages, and they are activated. They produce cytokines, and that's what drives the systemic inflammation. And then there's this gut lymph concept, which was published by Edwin Deitch in 2006. he never made any attempt to take that concept through to treatment of patients. It was all done in rats. But I can't take primacy there. But we did publish this paper independently of him and came to the same conclusion that the lymph system that drains the gut might actually be a way in which badness, to use a very medical term, badness might actually get through to the lymph, and by getting into the lymph can get into the circulation, and by getting into the circulation can actually impact organs. Just a couple of words about lymph, because not everybody is aware of lymphatics, and we've made a special study of that. We weren't there in 1670 where the groin lymphatics were demonstrated by this Dutchman. But you can see lymph nodes, opalescent lymph nodes, and it's a bit of artistic license here, mind, but you can see the little lymphatics joining the lymph nodes together, almost like a pearl necklace. So when we teach in intermediate schools, when we go along, everybody knows that arteries are red, veins are blue, lymphatics are green. Well, they're not, but that's how we depict them. And the lymphatic system is depicted here from the Cleveland Clinic. And you can see that the capillaries of the lymphatic system are little like fingers that sit amongst the cells. Fluid gets into them and are passed up through capillaries, lymphatic vessels, lymphatic duct, through nodes, actually traverse through the nodes, and then actually end up in the main collecting duct, which is called the thoracic duct. And we're going to talk a little bit about the thoracic duct. Interesting about the thoracic duct is that the lymph that runs up there bypasses the liver. It doesn't go through a detoxification system or get sorted by the liver as the blood does. Lymphatics bypasses it. And it does that because lymphatics absorb fat, and you don't want a lot of fat going to the liver. At least that's my simple explanation. I'm only a surgeon. So the thoracic duct enters into the circulation, and as we'll see in a minute, that's unfiltered, not detoxified badness that's getting into the circulation. The roles of the lymph in normal health is that we absorb fat by that means. That's how we get our fat when we eat fat. It returns fluid from around the cells, what's called the interstitial space. If you didn't have the lymphatics, you would just blow up and pop. I mean, this is the way that we return fluid to the system. And you need fluid to get out of the system, to bathe the cells, to give them the nutrients, to take away the poisons from the cell. So that process of exchange is really important. But if you just left the fluid there and didn't have a lymphatic system, then you wouldn't live. It's vital to life. And then, of course, immune cells traverse the lymphatic system, are really important for lymph surveillance all over the body. So just coming back now to the gut lymph model, we tried to summarize it in this paper. Let me take you through it just so we're very clear about that, because so much hinges on this in the rest of the talk. Steal my thunder. Okay, guys, here we are. Number one, those are the lymphatics in the gut. So if you've got gut injury from all those reasons we talked about, badness gets into the lymphatic system, correct? In the lymphatic system, it goes up to a little receptacle, if you like, called the cisterna chyli. And then the cisterna chyli becomes the thoracic duct, number 3, bypassing the liver, going all the way up and entering into the subclavian or the internal jugular vein on the left, not the right, just on the left. And where does it go from there? It's directly exposed to the heart, the lungs, the kidneys. Those are the organs which fail most often, which kill people most, and yet we haven't really connected the dots until very recently. And when I teach about it, I kind of talk about it. Really, it's like unfiltered sewer just getting dumped into the circulation. That's what it's like. Okay, that concept is based on some experimental work. I'm going to make this as painless as possible for you, but I do think you need to see a little bit of science, a little bit more modern than that. So the first thing is, can we show that the composition of lymph coming out of the gut in these circumstances is indeed changed? The composition, are things different with that lymph? Because we're making a claim that it might actually drive organ failure. And so we we've done that, and we've looked at multiple ways of looking at compositional change, and I'm not going to go through all that other than to say that talk about proteomics. Proteomics is looking at what proteins are in the lymph and what changes in proteins occur with acute pancreatitis. So what we have here is all of the protein groups. You'll see there's pancreatic enzymes, just 2%, and protease inhibitors. The largest protein group is the group which is protecting the body against the action of proteases, which damage protein. And of course, the enzymes from the pancreas are about breaking down protein. So pancreatic enzymes only represent 2%, but the protease inhibitors, 16%. It's almost like there's protection there waiting for something to happen. So that's in normal. What about the proteome from lymph in acute pancreatitis? Well, it really surprised us. Of all the different proteins that are in lymph, only 8 proteins we were able to demonstrate increased their levels in acute pancreatitis. That's only 3% of the proteins. And when you actually look at them, 7 of the 8 proteins which increase in lymph are actually pancreatic-derived enzymes. This is lymph coming out of the gut, not lymph coming from the pancreas. And the one I wanted to draw your attention to the most is the one up here, pancreatic lipase, because that's going to come into the story in just a minute. So pancreatic lipase is 38 times increased in value, in concentration in lymph in acute pancreatitis. And there's no increase in the anti-protease protection. So the second experiment was to say that if this lymph coming out of the gut is nasty, what happens if we drop it onto some cells? Will they like it? Will they not? And so the cells we chose were a cell culture of cells that are derived from the lining of vessels, endothelial cells, hem sacs cells there. And what we've got here is a little graph, not too heavy, right? This is LDH, which is a measure of cell death. And what we've got here is control lymph, not very much death when you drop normal lymph. If you drop normal lymph and an anti-protease drug, not much death. If you have acute pancreatitis lymph on those cells, heaps of death. And then if you combined acute pancreatitis lymph with an anti-protease, back to very little death. So it tells us that there's, in acute pancreatitis, we've got something toxic to the lining of vessels. That's just one of many studies, but as an example of a compositional study that actually also confirms toxicity. I've already talked to you about the fact that in pancreatitis we get necrosis. Gee whiz, you'd think I'd have learned by now. Necrosis. And so one of the questions we asked was, is the lymph coming from the gut contributing to the failure of the circulation of the pancreas, which is responsible for the necrosis? You with me? So we had a nice setup where we had 2 models. Grateful to the rats for their help here. This is a little cuff that we put on the artery which supplies the gut, and we can blow up the cuff and make it ischemic. And the second model is we can put a little tube through the duodenum into the pancreatic duct and infuse terracolate, and that can give us a pretty predictable severe pancreatitis. And if we use a low concentration, a mild pancreatitis. Okay, so what we wanted to do was to take lymph, take lymph, it's far too complicated for me, take lymph from a rat that's been subjected to gut ischemia, not pancreatitis, just gut ischemia. So we've got lymph coming out of gut ischemia, and we then infuse it into another rat that's got mild pancreatitis. And we've got the abdomen open of that second rat so that we can actually look at a mild pancreatitis pancreas on a platform, warmed, heated, irrigated, and actually be able to bring a microscope down onto the organ and actually look at the circulation as we expose it to lymph from that intestinal ischemia. Is that okay? Okay, so maybe a bit too complicated, but here's what the capillaries, the blood supply, the little dots that are whizzing around are red cells actually. And so that shows you normal capillary blood flow. And then just what happens in acute pancreatitis starts to get slowed down, doesn't it? It doesn't whiz around quite so much. And so there's some impairment of flow there. And then when you've got acute pancreatitis plus lymph infused at the right concentration, what happens? Circulation pretty much shuts down. So that's a pretty potent something in that lymph, causing such a significant reduction in microcirculation. So acute pancreatitis, we know, can cause intestinal ischemia. Intestinal ischemia can actually impair pancreatic microcirculation and can make acute pancreatitis worse, as we demonstrated in that experiment with some other slides. So, the fourth experiment. So, this is gut lymph from acute pancreatitis causes heart failure. And this is it's always been a mystery to intensive care doctors, as far as I know, and I'm not one. What is the thing that depresses cardiac function? They've never actually found that. What's the cardiodepressant they talk about? Not the same kind of depression. Here we've got an acute pancreatitis model, the same one. We've taken acute gut lymph out of this rat's thoracic duct and infused it into a little rat heart, which is perfused and paced, and they actually use that for diabetic cardiomyopathy studies. So we're able to adapt it to this. And as a result of that, here's another graph. This is called the Starling curve, which is how we learn our cardiac physiology. But if you increase the load on the heart upstream, what the heart does is responds to that and actually puts more blood out. Makes sense. Put more in, get more out. If the heart's not working very well, you won't get so much out. And that's actually what's shown here, is that with look at this one here, the black one. There's a 20% reduction in cardiac output if you've got acute pancreatitis lymph infusing the heart at a really low concentration, which we think is the kind of concentration it would have normally. So this was almost immediate 20% reduction in cardiac output, cardiac failure as a result of lymph being infused into the heart. And to cut this one short, if you were to ligate the thoracic duct, so lymph can't get through to the circulation in acute pancreatitis, you can completely abrogate the heart failure. It just doesn't happen. So you can see that this is the cardiac failure that then returns to normal if you ligate the thoracic duct. So again, thoracic duct is a highway that is conducting a whole lot of badness from the gut to the circulation, and we've almost completely ignored it. So this is the last section. I'm probably ahead of schedule, am I? You're doing well. Yeah, okay. But we can take lots of questions. So that's the concept, that we've got gut lymph model says that damaged or injured gut in acute and critical illness produces badness, which is conducted by the lymph system to the circulation, and that causes organ dysfunction and then potentially failure. So in thinking about how to design translational strategies or treatment strategies, there's all sorts of things we can talk about. And I'm only going to talk about two of the several that we're working on. And I'm obviously quite excited about this. So finally, after, I think it was a TED talk in 2009, I first broached this idea. And now we're, many years later, finally getting translational strategies in place. So the first one is not original. It says, well, actually, if we put a tube into the thoracic duct and bypassed the circulation and allowed the lymph to drip into a bucket by the bed, if we did that early enough, would we actually be able to avoid some of the downstream effects of that bad lymph in the circulation on organ systems? So we actually did two reviews of the literature, one in English and one in Russian and Ukraine. There was a retired Russian professor in the University of Auckland who freely gave his time to translate 113 Russian papers, which we're really grateful for. Here's a really old picture from a surgical textbook showing that this is not something that's new. You look here, there's a tube going into the thoracic duct here, and here's a bottle by the bed with dripping dripping lymph. You can see the thoracic duct here entering the system, and they've got a tube sitting in there. So when we actually looked at the publications, one of the things that strikes you immediately is there's a flurry of activity in the 50s, 60s, 70s, into the 80s. The orange is the first paper, which was the English literature. The blue is the Russian and Ukraine literature. And you can see that the Russians and the Ukrainians did a few more studies more recently, but it completely died in the English literature. Nobody did it. Why? Well, one of the reasons for doing it was to actually cause immunosuppression in the early days before cyclosporin. And so they would actually drain lymph for up to 4 weeks to try and immunosuppress a patient before they did an organ transplantation. Cyclosporin came along, no longer needed. However, it was an open operation, and I think cosmetic reasons are very real. Nobody wants to have a cut in the neck like that. It's an invasive procedure. It needs a general anesthetic. But when you pull the tube out, not like arteries or veins, it doesn't clot. And so you end up getting a lot of lymph leakage, which we call a chyle leak or a lymph leak. And so that's a problem. Another problem really was when we looked at the quality of the studies. They're all pretty small. They're all underpowered. The statistics were pretty crappy. And you know, you'd have to say that when you looked at the results, the vast majority of them showed a positive result. And that's what surprised us, that nobody really had taken these early preliminary findings and actually put together a proper randomized controlled trial and demonstrated the benefit and actually introduced it into medicine. So I think the Forgotten Highway, this reference in case those hadn't guessed what we're talking about, I think the lymphatic is that, thoracic duct is that. There are good reasons to why we would want to access thoracic duct lymph. For us, it's gold, really hard to come by. The first one is therapeutic external drainage, which we'll talk about in just a second. But there are other reasons why. We could study the compositional changes that occur in lymph over the course of a disease. We can't do that at the moment. We could study the toxicity better than what we've done to date and do it in vivo by actually taking lymph from patients instead of from rats and actually demonstrating, confirming our rat findings. Really important is to identify potential drug targets. What is the toxicity factor? We've got a number of targets, but there are others which we haven't found yet. Let's find them and let's use combination therapy to take them out before they get to the circulation. We could actually design gut lymph targeted treatments, and we could actually measure the bioactivity in gut lymph if we could get a tube into the thoracic duct. So it's a precious fluid that we seek to study. So maybe a little bit brazenly, all done ethically and with the right approvals, we decided we need lymph. We need to study lymph. And as it happens, excuse the picture for those who are squeamish, this is a thoracotomy and opening the chest. I was doing an esophagectomy here. The lung is compressed above, ribs and intercostal veins are at the bottom, and in the middle is the aorta. This is the aorta. And running along the aorta is the thoracic duct with a little clip on it and a little cannula in it. So what we did with permission was, because we ligate or tie off the thoracic duct in patients anyway for cancer surgery, we thought we'd cannulate it for 7 days and then embolize it, meaning that the radiologist would go down the tube and just put a series of coils in there to stop so it doesn't leak. Unfortunately, 2 of my patients of the 4 did leak. And this was really hard to deal with. And we definitely discontinued the study. Actually, both of the reasons were avoidable. There's a lack of vigilance on the part of my research fellow for one of them. And the other was that the embolization step was delegated to a junior in the radiology department who put 3 coils instead of the 8 that's standard. So, you know, it was a pity. We did get some lymph and we've actually published on it. We've thanked the patients, especially the ones who had lymph leaks. They've come back to clinic. They're good with it, but it was a bit of a harrowing time, to be frank. And then we met a Russian, Professor Max Itkin. He works at the University of Pennsylvania, UPenn, in Philadelphia. And he's not cutting the neck. He's getting into the thoracic duct with a tube, without any incisions. And he's doing it consistently. I had a look at his data set of 962 cases, 2 episodes of minor SIRS response, maybe infection, but nobody had any major problems. I'm going to just show you his technique, which I think is a really major advance. It's not surgical, it's radiological. What you have here is a groin, and this is groin nodes. So what he does under ultrasound guidance is put a little needle into a groin node. You can probably feel your own, they're usually quite prominent, and then inject lepiodol, a contrast agent. And it's quite beautiful sitting there with the X-ray machine going to see the lymphatics go from the groin nodes around the edge of the pelvis, almost like a flower bloom, and then coalescing into the cisterna chyli, that receptacle at the lower end of the thoracic duct. And then the second step, which you see there, having opacified the thoracic duct, this is the scary bit for us when it started. I meant to bring it. I've got a needle which is literally this long with a little curve on the end of it, and he punctures the abdomen. I said, "What are you going through?" He says, "Well, I don't know, but it's 25 gauge and it doesn't seem to cause a problem." So he was very careful, and he just popped it in and then managed to decannulate the cisterna chyli, which is outlined with contrast, and then passes a wire up. I think I have a video that might work for you. Yes. Can you see a lasso there? And there's a wire. That wire is coming up the thoracic duct and then around there and into the vein. And then what happens here is he grabs the end of that wire, which has come up from below, and he pulls it out to the arm. So you've got a wire coming out the arm and a wire coming out the abdomen, same wire. And then over the wire on the arm, he can reliably pass a catheter into the thoracic duct. Wow. No incisions, no general anesthetic. He's got reliable access to the thoracic duct, and all of those reasons why we want lymph, we can now obtain by this method. Really a significant advance. And we took a radiologist from Auckland to UPenn, and they're now starting to do it here, which is great. So when you think about external drainage of thoracic duct, I think about, let's get rid of that toxicity, but then we could also get rid of excess fluid. Because if we can actually allow the thoracic duct to drain, all of that puffiness that patients are having, we might be able to unload the patient of some of the excess fluid. The only study that we could find in English literature that was done outside of Russia and Ukraine in acute pancreatitis patients was this one paper in 1989, which demonstrated improvement in respiratory function, circulatory function, and survival in patients who had external drainage of thoracic duct lymph in severe acute pancreatitis. I think it's a landmark study. Was never followed up. A proper full-size study hasn't followed it. They just simply said this could be a useful adjunct to the treatment of life-threatening complications in early phase acute pancreatitis. Needs to be repeated now that we've got a minimally invasive approach to the thoracic duct. So where are we at to? Well, number one is we set up a sepsis study in UPenn, because Max Itkin's there, he can do this for us, in an intensive care unit in UPenn. Screened over 60 patients during COVID. Nobody wanted to participate. So this is the reality sometimes. You set up a jolly good study and nobody wants to be part of it. So that was stopped, but I think for good external reasons. We've been back there and said, well, would you consider doing one in acute mesenteric ischemia? We talked about gut ischemia. In these patients, sometimes you have to revascularize, etc. So we've actually got a study with external drainage in patients having surgery for acute mesenteric ischemia. Those patients get sick afterwards. If we can stop the sickness, stop the organ failure, that will be an amazing study. Hopefully we'll see positive results. The other one, which is quite interesting, is that they have 23 cardiologists at UPenn, all dedicated to intractable congestive heart failure, meaning these are patients who are diuretic resistant. You know, they come in with heart failure and you want to get that fluid off their lungs, decongest them, but the kidneys won't work. In the 1960s, somebody did external drainage for patients with severe congestive heart failure, taking off up to 8 liters a day out the thoracic duct. Patients shrunk, patients walked out. That's amazing. And so that group have, because they're in the same hospital as Max said, kind of brought them together and say, look, you've got plenty of patients. Some of them are going to die. Well, most of the intractable ones will die without treatment. And maybe this is just a bridge to cardiac transplant. Get them home, get their nutrition better, get them in better shape, and then come forward for transplantation. So I think that's useful. And in Chengdu, China, we have a group of radiologists who are going to help do an external drainage in acute pancreatitis, which is what I'm really excited to do. I'm going up there next month to try and get that started. Okay, that's the first strategy, which is external drainage of thoracic duct lymph. Crude concept, just put it in a bucket. That's all we're doing. This one is much more sophisticated and beyond me, really. We're working with Monash Institute of Pharmaceutical Sciences, who are ranked QS #1 in the world for drug design and delivery, and they've been fantastic to work with. I'm not going to bore you with too much detail, but I want to come back to lipase. Remember that enzyme that went up a lot in the lymph? And so this here, over here, in yellow, which you can hardly read, says orlistat lipase inhibitor. Orlistat has been on the market for years as a drug to treat obesity. You may have heard of it. How does it work? Well, in the diet, if you have fat, lipase will come from the pancreas and break it down. If you stop the lipase from working, the fat stays in the bowel, and you get bloated, uncomfortable, don't want to eat, and you pass fatty stool. That's how it works. And it doesn't get into the circulation, it just stays in the gut. So the concept here is if we could use orlistat as an anti-lipase, and instead of it working to lose weight in the gut, we actually target it to the lymphatic system. Lipase working in the lymphatic system, we could stop lipase. Well, why would we want to stop lipase? Lipase produces, in its breakdown products, fatty acids, non-esterified fatty acids, which have been shown to cause respiratory failure and cardiac failure. So if we could actually have anti-lipase treatment targeted the lymph before lipase gets into the circulation, that's a target worth pursuing, which is what we've done. And so in order to do that, we've had to have these self-nano-emulsifying drug delivery systems, SNEDs, and you can see what goes into a SNED, the drug itself, a co-solvent, surfactant, and oil and lipid. And these form themselves into small bubbles, if you like. And as a result of that, instead of orlistat staying in the gut, what does it do? It gets into the lymphatics. And we've demonstrated that. Well, here it is. We tried to combine it with a number of different things. The one that seemed to work the best overall was actually oleic acid, a long-chain lipid. And we've demonstrated that it gets into lymph in high concentrations, which is here. And then here, we can actually show that orlistat in this situation also gets through to the plasma. And so here we've got a drug that gets delivered to where we want to get it to, but it's joined up with oleic acid. Well, the good thing is the target that we're going for is lipase, and lipase cleaves, this is pretty cool, lipase cleaves the tail, leaving lipase active, anti-lipase, orlistat active, beg your pardon. It cleaves the tail, leaves orlistat available to actually then counter the lipase, which is freed it up. And so it works, and I'll show you. So we set up a study where, a rat study in this case, of acute pancreatitis, said, well, if we give orlistat targeted to lymph, can we make a difference to the outcome of acute pancreatitis? What you see here, without going through too much detail, is you've got lymph treatment. If you give orlistat with a pancreatitis, you have toxicity to the cells. There's two different cell lines. This one you came across before. You can see that with the pink is with acute pancreatitis, and you have low viability. That is, not a lot of cells survive. If you give orlistat with acute pancreatitis in the way that we've just been talking about, the cells survive much, much better. So that's an indication that you can reverse the toxicity to cells with this approach. Lots and lots of pictures here. It makes a jot of difference to cytokines. This has nothing to do with cytokines. All of those on the back, you can see that the blue columns are pretty similar to the pink columns. There's no real difference. And then the next part of this was plasma lipase activity, that is lipase that's come up the thoracic duct into the circulation, and free fatty acid levels. And what you can see is that if you give orlistat with acute pancreatitis, lipase levels in the blood goes down, good sign. And what you see is that the toxic free fatty acids dramatically decrease. And so that if it's true that fatty acids drive organ failure, this is going to be helpful. Well, we went further than that, and we looked at actually what happens to blood pressure, because with acute pancreatitis, the blood pressure sagged, and in fact, the orlistat restored the blood pressure. You can see that there, restored blood pressure. Then we looked at markers of heart function. There's a whole lot here, CK, MB, troponin, fatty acid binding protein, etc. And what you can see in most of these is that acute pancreatitis levels are high, and we actually improve the levels. All of these different markers of cardiac function improve with a treatment of anti-lipase therapy to gut lymph. Amazing. We went further than that, and so we wanted to look at other organ systems. Here you've got kidney markers, creatinine and urea. Here you've got liver markers. The same picture all the way along. Orlistat is beneficial in every single case. And this is at 3.5 hours after the induction of pancreatitis. So, coming to the end. We're very excited about that animal study, and I don't think I've shown it here, but we are now moving ahead with that drug formulation, now patented. Actually, we've got it here. We're looking for a partner to help us. We're planning the first human randomized control trial in Chengdu. I'm going to be dealing with that when I go up there as well next month. IP secured, commercial partners being sought to allow us to proceed with that. But this is the first instance of a lymph-targeted drug impacting systemic inflammation and end organ function. And the exciting thing is that the orlistat is already FDA approved. The excipient, which is all the rest of the stuff, is also FDA approved for a different indication, mind you. But we're a long way towards getting FDA approval. We don't need it for a research trial, but we're underway with that. So for the first time, we may well have a treatment that's effective. So in conclusion, we've talked about organ failure being the most common cause of death from acute pancreatitis and most acute and critical illness. No specific treatment for organ failure, really good at organ support. The role of lymph has largely been forgotten. Gut lymph model is a new paradigm which is translating now into two different research or translational therapeutic strategies for us, external drainage and gut lymph targeted drugs. And we've got clinical studies in the US and China underway. I can't not acknowledge AMRF here because this support, I cut out the amount of money, but it was $159,000. Really timely support for us in gut lymph targeted treatment, severe acute pancreatitis. Not the only funder, but what AMRF has done for me over the years is actually evened out the lumpiness of funding. We might get a project grant from HSC or a program grant, but they end, and you often have these valleys, doom in between major grants. And AMRF has kept us alive on more than one occasion, almost like organ support. It's been amazing. So I'm really, really grateful to AMRF for that. There's lots and lots and lots of people who've been involved in these studies and others. And you can see the Monash University figuring very largely there. So thank you very much.
And John, that is for sure. Well done. Yeah, absolutely. So look, I think we'll do 5 minutes for questions because I know that people are probably wanting to go and, yeah, understand if you don't have any questions at the moment, but we've got a question.
I've got 2 questions. Does a diverse and healthy gut microbiota have a protective effect for this inflammatory response? And the second question is that drug therapies which are used for localized lymphatic tumors, do they have a benefit in this lymphatic inflammatory pathway?
I don't know the answer to the second one, but the target there would be the vessels. And what we're wanting to is improve lymphatic function, because you want to take out the lymphatics as a conduit for metastases, right? So if anything, you don't want good lymphatic function around a tumor, but you do want it for the other reasons of getting rid of badness. But so no, I don't know whether that would work or not. I'll give some thought to that. The first one was microbiome. Yeah, there's a huge amount of work going on all around the world about this. The sad thing in acute pancreatitis was that there was a publication, the Propatria trial in the Lancet, probably 10 years ago now, where they had identified several probiotics that they thought were beneficial. Randomized control trial, beautifully done by the Dutch multi-center, 20 centers, and there were 8 deaths, and they were from gut ischemia. My explanation for that is that they were too aggressive with ventral feeding, putting a huge metabolic demand on the gut, irrespective of the probiotics, and got gut ischemia from that. We now don't do it that way anymore. Is there a role? Definitely. Do we know which parts of the complex microbiome are beneficial? Starting to understand that. How we might use that therapeutically, not established yet, but I'm sure it's going to be an important component of treatment in the future. Just not there yet. It's complex.